Dear student, welcome to the course of Basic Genetics. I am Richa Arya and today we are going to start a new unit in a new week. So, today we will be discussing about the gene mapping on eukaryotic chromosomes. And in today's lecture, I will discuss about linkage and crossing over. So, these are the concepts that you should understand because these concepts help you build the idea how people actually end up understanding more about genes and chromosome and their order. So, if you remember, we were initially were talking about Mendel's work, and we said that Mendel, people say that he was really very lucky that he ended up looking at 7 traits that were independently assorting, but people later on they were not really finding those clean results with the genes that they were studying. But that is how we discovered things. It is nice that we have some simple principle discovered probably early on, and then we have end up adding complications. Otherwise, we will be so puzzled with all the complicated and all sort of different behavior that it will be very difficult to really conclude something meaningful out of that. So, now coming back to today's context is the work, the experimental work done by Batterson, Saunders and Punnett. So, what they were doing, they were also trying to understand the genes, right. It is the 1900 when those the Mendelian principles were already discovered, and people were also trying to understand genes in different organisms. So, these people were working on sweet pea. Remember, Mendel was working on garden pea, and these people are working on sweet pea. And what are they focusing on? They were focusing on flower color, which was purple and red, and they are looking at the pollen shape. Remember, 2 characters at a time when you want to study the independent assortment. So, for the pollen shape they were having long and round. So, these are the 2 major character classes that they were focusing, but to their surprise the F1 was fine, but when they did the F1 self cross they did not find the 9:3:3:1 ratio. So, if you see, this is the data from their crosses. So, this is just to remind you that they were trying to understand the independent assortment in the garden pea, and they were looking at flower color and shape. So, purple and long, I already reminded you, you can go to the week 1 classes how these genotypes are written. I will briefly tell you some more facts about these genotypes, but in my week 1 class I did mention when we put this bar, what does it mean, right. So, this bar actually tells you that it is a dominant, phenotypically it is dominant, but and that is why we are putting a capital letter here, but then here it could be capital or recessive, we do not know until we do more crosses. So, you can see that for purple and long, let us look at the expected first. So, just look at this column. This shows that if you have 6,952 phenotypes that you have observed, the organisms that you have observed, the plants that you have observed independently, what should be the distribution in 9:3:3:1 ratio for purple and long and purple and round and red and long and red and round. So, this should be the ratio that you expect. But what they were seeing, there are 2 major classes that were predominant, right, and there are very few classes that were recombinants, right, that were the new combination. They really look like parentals, and there are very few that were non-parental combinations. So, they did observe, but they did not really understand, or they really do not explain what could be the reason for this exception, right. So, it was there, this observation that things deviate from Mendel's rule was discovered, law of independent assortment, but their method, the mechanism was not discovered. So, this is again we discussed about Morgan, because Morgan was one who identified white gene, and he showed that the genes are located on chromosome. His lab has enormous contribution in genetics in terms of identifying genes and mapping them on the chromosome and their functions. So, he did a lot of work on genetics using the Drosophila or fruit fly. So, now when Morgan started working on the fruit fly, what he found when he was studying the autosomal genes, I will show you in a minute. So, what characters he was using? He was using eye color. So, he has purple PR, this is a symbol for the purple gene, and this and the red eye. So, remember there is one thing that you should just remember how the genotypes are written. When we say plus on anything, we are saying that it is wild type, normal, and if we are not putting anything, then it is just recessive. And you would also see there are many numbers or letters that people put on as a superscript, and that also indicate different type of mutations in it, right. So, that is the way how we write the genes and different alleles of a particular gene, right. So, what he was looking at? He was looking at eye color, and the second character that he was looking at was wing length. So, in wing length there was one wing which was normal and another one was vestigial. So, actually unfortunately I could not find a good image for purple. I can show you how a normal wing in fruit fly looks like. It is like this, a very straight, flat, beautifully laid on each other, and the vestigial wings are very small, they are very rudimentary. You can see that they are very underdeveloped and easily identifiable, right. So, this is a recessive trait. Recessive means when this trait is homozygous, you see this phenotype, right. So, what did he do? In the parents, he have these parents where purple and vestigial. So, one more thing, now let us see what this slash means. So, you see we can write genes like this, PR, VG, then one line showing one homologous chromosome, another line showing another homologous chromosome, PR, VG. Or we can also write it like this for simplicity, right. We are not using two lines, we are just using one line as a slash, and we can write them, you know, this way. So, remember there are certain different ways to write these genotypes, but once you understand how to read these genotype, it will be easier for you. So, what we are seeing here? We are seeing here purple and vestigial and wild type for eye color and wild type for wings. And in F1 generation what you get? You get heterozygous, right. You get both the alleles for both the genes, and when you set up a test cross of this with the recessive parent, right, then you expect a combination. So, I hope that you remember the Mendelian test cross ratios are 1:1:1:1. But Morgan's test cross ratios deviated. So, you can see he again saw this purple vestigial, which was the wild type, a lot of them. The recessive ones, again a lot of them, whereas these two are very little, where you have wild type eye and vestigial wing, and you have purple eye and wild type wings, right. So, this ratio deviated again from Mendelian ratio, as we saw in case of sweet pea, the experiment by Punnett and team. So, because this ratio deviated drastically from 1:1 test cross ratio, Morgan started looking into why, right. So, what he did is, now just pay one attention that here in this cross they are homozygous, right, purple and vestigial or wild type for wing and eye color. Now, what he did is he used this combination, the recombinant. We will talk about recombinants little later in more detail, but just see there is one gene which is normal and another is mutation, or one gene which has mutation or another is normal, right. So, he took this as a parent, right, these heterozygotes, right, and then he crossed them with another type of heterozygote, and he has this F1 generation where he got the combination of purple and wild type and purple and wild type and vestigial, right. So, here wild type eye color, vestigial wing, here purple eye color, wild type wing. So one mutation, another is normal. We'll talk about it in our next class when we will talk about coupling and repulsion, but just so understand that what he is trying to understand, when he has a different combination of two genes with their alleles, what is the outcome. So if you see, when he has this progeny, and when he put the test cross with the tester male, recessive male for vestigial and purple, what he found, you see, now the major class here, they are the parental class. Before also, whatever the parental genotype was the dominating class, whereas the new classes are less. You see, now both the genes wild type and both the genes recessive, they are less, because they are the new combinations that has to be formed. So he clearly saw that in these crosses, what is happening, the parental genotype is dominating. And so Morgan suggested that the two genes in the analysis are located on the homologous pairs of chromosomes. So he suggested, remember that was a suggestion, and you will see how he has proven that it is indeed the fact. So he considered, so remember before they were also putting this dot here. So when we really do not know the position of genes, we just put a dot to say that they are on, it looks like that they are on the same chromosome, or maybe different, we do not know, but we are trying to understand the interaction between two genes. Once we know it clearly, then we neatly put the genes on the lines as I have shown you before. So here you can see that he placed these two genes on one homologous chromosome, and this is another homologous chromosome, and in a cross you get these gametes, and in F1 generation you get the heterozygous progeny. So genes on the same chromosome, he considered them linked. Remember this point, right? So he was saying that when two genes are on one chromosome, we call them as linked, right? But then the question is, if they are linked, then how can you expect them to separate even in a smaller ratio? If they are linked, they should be linked, right? We should not get any recombinant. So to explain this, Morgan proposed the theory of crossing over. So he proposed, and in his lab he saw how actually crossing over happens, and it was also discovered independently. We will cover all those details later, but let us now look into the idea that first he proposed that two genes can be present on one chromosome, and when they are present on one chromosome, we call that they are linked. And now he is proposing, if they are able, if they manage to separate, then this is happening because there is a phenomena of crossing over is taking place. And so the crossing over is the phenomena where the fragment of homologous chromosome break and exchange. So this is just the same parental combination as I have written before, and you can see in the F1 generation you get the homologous chromosomes with heterozygous combination, and if the crossing over is taking place, right. So this is just to show the two chromatids on one chromosome and the two chromatids for this brown chromosome, and if one of the chromatid of each chromosome is doing this exchange, then what you end up seeing? You end up seeing the, so if this sister chromatid gets separated, you get the parental combination for both the cases, and here you get the exchange, right. So you get this exchange where you have the wild type I and vestigial wing and vice versa. So these are the two recombinant classes that you get. So chromosome breaking and physical exchange of chromosome segment to make non-parental combination is the crossing over. So now the question that arises is that crossing over occur at which stage? We know that chromosomes replicate in S phase and then they divide in M phase, right. So when exactly this crossing over is taking place, before replication or after replication? So you see that people have done these experiments to understand if at what stage actually the recombination is happening, and you can also appreciate it when you look at the analysis. Let us assume that the chromosomes exchange, the crossing over is taking place when they are the 2 chromosomes. So, before replication, when the DNA amount is still not doubled, right. So, what you see, if the exchange is happening at the 2 chromosome stage, then you have already the recombinants, and then these recombinant chromosome will replicate, and then these replicated sister chromatid will separate it. So, in this case you will never have any parental genotype, right, which Morgan did not see. But if the recombination is happening at the 4 chromatid state, then just look at it, what will be happening. So, if this is the again 2 chromosomes present, and then these 2 chromosomes exchange the segments, one sister chromatid is exchanging the fragment, the same as we have discussed before, which means the replication has already taken place. One chromosome now have 2 sister chromatid, and then you will have one sister chromatid which is like parental type in both the chromosomes, and one of them is the recombinant type. So, in this kind of condition you will have the parental type as well as recombinant type. So, because more than 2 different products of single meiosis can be seen in some tetrad, I will discuss about tetrad in another class just for the sake of simplicity, but just understand this concept, how exactly we are seeing recombinant if it is happening, if at all it is happening in 2 chromosome stage or 4 chromatid stage. The crossing over cannot occur at 2 chromosome stage. So, for any pair of homologous chromosome, now what we want to know, we already learned that okay, crossing over is taking place when there is already replication happened, when we have the sister chromatid present, fine, right. Now we want to know how many sister chromatids are actually be involved in this process. Is it always, like last time also I said that 2 sister chromatids are involved, is it the only case or there are other possibilities? So, 2, 3 or all 4 chromatids can be involved in chromosome in the exchange of the fragment. It is not limited to one or the other, right. So, remember that. So, this is a drawing where I am showing you the involvement of 3 chromatid. You see this first chromatid is getting exchanged with this sister chromatid. The another sister chromatid of this chromosome is also getting exchanged with the same sister chromatid, right. So, in total there are 3 sister chromatids that are involved. So, in that case what you are getting? You are getting 3 recombinants and 1 parental type combination, right. And if you see, if you get all the 4 chromatids involved in the crossover, in the double crossovers, then you see this one is involved with this one is involved with this one, and in this particular case you are getting all the recombinants, right. So, by getting the counting the recombinants and type of parental type, you will be able to understand up to what extent the crossing over is taking place. So, with that I have shown you the importance of exchange of chromosomes. You see, independent assortment was one of the way that Mendel told us that generate new combination. Now, Morgan is telling us another way through which new combinations can be generated even when the genes are linked. Although the new combination may arise in lesser number, but they do appear, and so it is important for the variations are always important for evolution. So, this is a little complex topic. I would highly recommend you to read little bit more about it, and if you have any questions, do approach me and I will help you out as much as possible. I will leave you with these 2 questions. The first question is a question that you need to solve, right. So, if a double crossover occur involving only 2 chromatid, remember only 2 chromatid between 3 genes, which of the following combination cannot be produced? So that is what you need to look at and let me know which are the combinations that you are not expecting to see, and I want you to answer in detail so that I can understand if you get the concept correctly. And the second thing is, I want you to look at this genotype that somebody has written and let me know if these genotypes that is written here is correctly written or not. These are the two homologous chromosomes showing two genes. Do you think the way somebody has written it, or who have designed the question has written it, do you think it is right or wrong, and justify your answer. So with that, I thank you all for your time, and again, if you have any questions, please do write me or write in the chat box. Thank you.